Good day everyone. Welcome to the course on next generation sequencing technologies, data analysis and applications. We are into the final week of the course and we have discussed the genome sequencing part, identification of single nucleotide polymorphisms and transcriptome sequencing. What is remaining is the application of NGS technologies in epigenomic studies, and this is what we will be discussing in this class and the last few classes. So, these are the topics that we will be covering today. So, we will mostly talk about epigenetic modifications, because it is important to understand what modifications we are looking at or what modifications we want to identify using the NGS methods. We will talk about epigenomics, and then we will talk about or briefly introduce this application of NGS for epigenomics, and in the subsequent classes we will actually expand on this application part a lot more. So, these are the keywords for this class: DNA methylation, histone modifications and nucleosomes. So, going back to the flowchart for NGS data analysis, we started with the read data from sequencer, and this is common for all applications, right. We looked at different data formats. The first step is the quality control, we talked about that, and then we talked about read mapping process, and we applied that this read mapping process for identification of single nucleotide polymorphisms, indels, structural variants. We also talked about transcriptome analysis. Again, mapping is the first step in that process. On the other side, we talked about read assembly in last few classes. So, last week we talked about the assembly algorithms, we talked about tools, and we then get a new reference genome after the assembly process. So, we have discussed all this, and what you see, the epigenomic part is remaining, and this is something what we are going to discuss in the next few classes. Okay, so, the question is, why did I left this epigenomic studies for the last part, right? Why didn't we discuss after the transcriptome analysis? Okay, so, I will start with that. That's because this studying epigenomes, it actually traditionally requires specialized treatments and processing of the genomic DNA. Okay. So, all types of applications that we talked about, there is some sort of experimental step, right. So, we isolate DNA, we then fragment DNA, we add adapter, we then do amplification, etc. For RNA-seq, we again convert to cDNA, etc. Then prepare the DNA library for sequencing through adapter ligation, etc., amplification, and then do the sequencing. For epigenomic studies, the DNA has to be treated chemically through different chemicals, right. So, this is something we will discuss again, which means it adds to the complexity of the whole process. Okay. So, this is unlike DNA or RNA processing, which still have some experimental step, but these are simple steps. Epigenomic studies, they require these specialized treatments, which means we need to have controls, and this makes the experimental process more complex, but also the data analysis process more complex, because you need to have more control experiments now, okay, to account for these different biases that will be introduced during the experimental steps. Okay. So, this is the reason why I chose to keep it at the end, because we will also need to talk about the experimental steps and their implications on the data analysis. So, let us now start with the epigenetic modifications, right, and what are epigenetic modifications, what are the different types of epigenetic modifications, that is something I will introduce. Okay. So, epigenetic modifications are modifications on the genomic DNA that are not associated with any change in the DNA sequence. Okay. So, these modifications would not change the DNA sequence, right. It will not change this ATGC base, it will simply add on something to the genomic DNA, right. So, it will add something extra to the genomic DNA, and that is why it is called epigenetic, right. So, its features on top of genetics. Okay. So, it is not changing the sequence itself, it is adding something on top of the genetics, on top of the genomes. Okay. So, there are different types of epigenetic modifications, and the first one is very common one is called DNA methylation. Okay. And this is something you probably all are aware of, right. So, these are methyl groups that are added directly on the DNA base, right. So, if you have the DNA strand, right, you have these methyl groups added to the bases, and most of these additions happen on A or C base, right. This is commonly studied, and we have seen this in prokaryotes as well as in eukaryotic systems. And also what we see now is that these are not just methyl groups, there could also be hydroxymethyl or carboxyl groups that are added on top of these bases. Okay. We will talk about this in much more detail in the subsequent class, right, where we will actually specifically focus on this DNA methylation. So, it is important to know these modifications, because then you can devise strategies to identify these modifications using the next generation sequencing methods. Okay. As you can see, without chemical treatments or experiments, right, we cannot directly detect this, okay, at least with the traditional next generation sequencing methods, but the newer techniques, the single molecule sequencing methods, they can actually help us identify them directly. Okay. So, we will talk about this again in the next class, where we will talk about how we will identify these modifications if we are using the Illumina platform, or how we can identify these modifications if we go through the nanopore or the SMRT platform. You can also have something called histone tail modifications. Okay. So, histones are proteins that help in packing of DNA in the nucleus, and these DNA actually wraps around histones without going into too much detail, right, all those nice figures you probably can see in the textbooks. And you can have these histones being modified at certain residues. Okay. So, you can have methylation, acetylation, etc., on the histone residues. Okay. And there are specific type of modifications which I will just mention one or two of them. For example, histone 3 lysine 4 residue, this is modified, histone 3 lysine 36, these are modified. Okay. So, again there are specific bases, specific places or specific residues of histones that are modified, and you will see methylation or acetylation or even other types of modifications. Now, what happens is that when you have this chemical modification of histones, they affect binding of histones to DNA. Okay. So, these modifications are very important for controlling this binding of DNA to the histones. Okay. So, how tightly this DNA will bind to the histones will be affected. You can also have something called chromatin remodeling. Okay. So, chromatin remodeling, okay, so this means there is change in the chromatin structure. Okay. And this is because the alterations or changes in association of DNA with histones. Okay. As I just told you, right, DNA wraps around histones. Now, if you have changes in this association, right, if that histone is coming off from the DNA or the histone is going in and binding to the DNA, right, that will actually lead to change in the chromatin structure. Okay. And this is what we call as chromatin remodeling. Okay. And this can also happen due to sliding and shifting of histones in the DNA, right. So, you can have association, dissociation, you can have sliding, shifting, etc., and this is usually done by some proteins which are called chromatin remodelers. Okay. So, again without going into the details, I will not mention the names of those, but then again these proteins exist in all organisms, all higher eukaryotic organisms, for example, in humans also. Okay. So, now we understand these different types of epigenetic modifications, and we would like to identify these modifications, right. So, that is why we discussed, right, what are the different types of modifications that will be targets for our NGS methods, right. Now, as I said, like we want to talk about epigenomes, right. So, the question is, what is epigenome? So, epigenome is simply the ensemble of genome wide epigenetic modifications, right. So, we have these different types of epigenetic modifications, and what we want to do when we are studying epigenome is we want to look at all these types of modifications across the whole genome. Okay. So, if you take the full genome and you want to identify all these modifications that are present in there. Now, one of the things you probably realize is that these modifications, these are actually transient. Okay. These are not always permanent, right, that like DNA sequence, that it will be there and the base will not change. Okay. So, these modifications are transient, they can change, as we just mentioned, right. You can have this chromatin remodeling, so again this is changing with time, and similarly you can have these histone modifications or DNA methylation that can also change. Okay. You have proteins or enzymes that will add these groups, and there are proteins and enzymes that will remove these groups. Okay. So, again this actually complicates the full analysis, right, because there is a time component here. Now, also epigenome is sometimes broadly defined. So, this is also connected to the gene expression state. So, as you can realize and we will discuss a bit later, is that all these modifications on the DNA, they have some impact. Okay? It is not like they are just sitting there doing nothing, they have some impact on the expression state of the cell. Okay? So, this means there is a connection between epigenome and the transcriptome. Okay? So, by looking at the epigenetic modifications, you can sometimes say which gene will express at high level or which gene will not be expressed. Okay? And we can actually study these connections if we look at the epigenomic data and transcriptomic data from the same cells. Okay? So, that is why epigenome is actually connected with gene expression state, and it can be simply the global gene expression state that is not associated to mutations in the genome. So, if you take mutations out of the picture, so because if you have mutations in the promoter regions of a gene, this will affect the gene expression state. So, if you take out the mutations, whatever changes are happening in global gene expression state, that will be because of the epigenome. Okay, so what is epigenomics is simply the study of these epigenetic modifications at the genome scale. As I mentioned, right, if you look at human genome, right, 3 billion bases, we want to look at all the epigenetic modifications across the full genome. Okay, so this is a genome wide study and not limited to just local regions or specific genomic segments, right. So, it has to be high throughput in nature, and this is where the next generation sequencing technology is coming. Okay, so the question you might ask, right, why do we study epigenomes? Okay, so there are two parts, right. So, of course, why do we study epigenomes, and then the next part is how. Okay, so how part we will just introduce in this class, and in subsequent classes I will discuss them in detail, right. How do we apply the next generation sequencing methods to actually understand or to study these epigenomic variations. Okay, so the question is, why do we study epigenomes? What's the impact or importance, right? So, as I have just mentioned, epigenetic modifications impact gene expression state of a cell, and this is we now know, and these combinations of these different types of modifications can have different unique effects. Okay, so we talked about 3 different types of modifications, right. So, we talked about DNA methylation, histone modification, and chromatin remodeling. So, you can take any combinations, and these combinations will have unique impact on the expression level, right. So, this is what we want to understand, right, how epigenomes or how epigenetic modifications across the genome impact the expression levels of the cell. Okay, so, this is something now we realize, right, this is actually very important for gene expression, and I will just illustrate with some examples how actually these modifications impact gene expression. So, let us take DNA methylation first. So, these are direct modifications on the DNA basis, and one of the very common modifications that we know is on C base in something called CpG island. So, these are CG bases that are present in promoter regions of genes, especially in mammalian systems, and methylation of this C base in this context, it actually leads to silencing of gene expression. Okay, so, this is something that is well known. So, if you can study this CpG methylation, you might want to you might be able to predict, right, which genes will be silenced in a cell. Similarly, if you look at histone modifications, and this has been studied by researchers, that they are also associated with different aspects. Okay. So, I have just mentioned, right, there are different residues of histones that are methylated or acetylated. So, here are 2 examples. For example, you have H3K4Me2 or H3K36Me3, right. So, H3K4 is the histone 3, lysine 4, Me2 means you have double methylation, right. So, dimethylation, and K36Me3 36 lysine, you have triple methylation, 3 methylations. Okay. And you can have all sorts of combinations. There are other residues which are also commonly modified, and they have been associated with specific gene expression patterns. So, some of these modifications are associated with activation of gene expression, and some of them are associated with repression of gene expression. Okay. So, again we do not have to remember all these combinations. If you want to understand the biology, or if you want to predict the biology, right, then you will remember, okay, this methylation pattern or this modification actually gives rise to activation, or this modification pattern leads to repression, right. Our goal is to identify these modifications, right, and to see across the genome, right, through the application of next generation sequencing technologies. Okay, so as I just mentioned, site of modification determines activation or silencing of gene expression. So this is something we want to study across the genome and then perhaps relate to transcriptome or gene expression levels. And what has been seen is that again we have these different combinations of histone modifications, right. So, you can have HTK4-ME2 with something else, HTK36 ME3 or some other combinations, right. Again there are many sites which can be modified. You can have dimethylation, trimethylation, etc., and you can generate these different combinations, and there are unique effects of these combinations. Okay, and this is something that actually referred to as histone codes. So, you can probably, once you study this, right, once you associate these modifications with expression, you can then perhaps predict, okay, with this combination what would be the expression state of a gene. The third part which we talked about is the chromatin remodeling part, okay, and this is related to condensation or decondensation of chromatin. Again here this is because of the nucleosome. So, nucleosome is the DNA bound with the histone, so that gives rise to nucleosome, and where you have nucleosome, right. So, that will be more condensed, and this region is unlikely to be active, right, because for expression, for transcription to happen, a DNA has to be free, right, with the scope of the transcription factor binding, RNA polymerase binding. But if it is kind of compact already because of the nucleosomes, right, that region is unlikely to be expressed. Okay, and that gives rise to two regions, two different types of regions we called heterochromatin and euchromatin. Okay, so what is heterochromatin? This is actually condensed form. This is bound by nucleosomes. So, you have all those proteins in there. So, this is in compact condensed form, and this is transcriptionally silenced. And on the other hand, we have the euchromatin, which is in the decondensed form, or sometimes we refer as open form, okay, and which means it is free to bind transcription factors and RNA polymerases, and this is the transcriptionally active region. Okay, so as you can see, we are learning a lot of concepts, right. So, different types of epigenetic modifications, and we are learning how these modifications, right, how they can impact gene expression. Okay, so this is the idea that we want to take forward from this class, right. So, these modifications are important and they actually affect gene expression, and I will also finally also give some examples of how epigenetic modifications are also important for different phenotypes and behaviors of cells and organisms. There is another part, another type of modification which we have not talked about. This actually happens because of the 3D genome configuration. So, this is something that is very new, that has been studied, being studied by researchers. We still do not have the complete picture of this, right. So, what do you mean by 3D genome configuration? Okay. So, 3D genome configuration means you can imagine the DNA, right. So, it is in the nucleus, it also has a 3-dimensional structure. Okay. And it is not like everything is compact and everything sitting in one place, etc. This genome has a 3-dimensional structure, okay, and this structure can impact the gene expression pattern. Okay. So, this is what the researchers have seen now, is that there are regions called chromosome territories. So, if you are talking about an organism, right, where you have, let us say 16 chromosomes, right, each chromosome will occupy a certain space inside the nucleus. It is not like they are all jumbled up together, they are separated and they take their own space. Okay. So, this has been now studied. So, and there are also something called AB compartments, there are other different components of these, okay, and we will talk about them when we talk about this 3D genome configuration and how we can study this configuration through application of NGS technologies. Okay, so, what you see now is these applications, the epigenomics actually is a huge field. It encompasses different aspects. Okay. So, it starts with the modifications which we talked about, DNA methylation, histone modifications, but it also is associated with the nucleosome occupancy. It is not a chemical modification, right. It is actually something just association of histone with the DNA, and here we are simply talking about the 3-dimensional structure of the genome, right, and that can also have impact on gene expression. Why do you want to study epigenome, right? What is the importance of epigenome, right? And one of the things that we see is that this actually these epigenetic modifications, they actually give flexibility in cellular phenotypes and behavior. Now, if you think or compare against genetic mutations, okay, so the single nucleotide polymorphisms that we discussed, those are mutations that actually change the base, right. So, that is actually is going to happen mostly in one direction, right. See, it is very unlikely that you are going to revert back to the original sequence, that rarely happens in nature. Whereas, if you think about these epigenetic modifications, these are reversible. Okay. So, all these histone modifications or the DNA methylations that you see, right, those can be added and removed because you have all this machinery inside the cell that will do this. Okay. So, you can add them and remove them, right. So, it gives cells flexibility, right. So, if they want to express certain genes or if they require expression of certain genes, they can remove certain marks and that can help in expression of that gene, or if the cell does not require expression of certain gene, it can also silence through this epigenetic modification, right. So, as you can see, this gives a good flexibility in terms of cellular phenotypes and behavior. So, you can modify gene expression according to the need. Okay. So, and I mean clear enough, right. This what you see is that most of these epigenetic modifications, they happen, right. They are actually changing in response to the environmental conditions or cellular signals, right. So, in case of humans, right, of course, this is also affected by, for example, not just environmental conditions, our diet, right, lifestyle, etc. So, everything actually plays a role in epigenetic modifications, right. So, this is again giving us the flexibility, right. So, you have this flexibility in cellular phenotypes and behavior. So, just to illustrate this with a figure, so you can have a cell that can actually exist in two states, right, and this can be driven by this epigenetic modification. So, you can have state 1, state 2, this is reversible, and this is driven mostly by epigenetic modification. This gives the cell this flexibility. Now, this flexibility has very important implications throughout different processes in biological systems. Okay. So, this flexibility is important for importance in cellular decisions, right. So, if you probably have heard about stem cell differentiation and pluripotency, right. So, here you have a important role of epigenetic modifications or epigenome. Then you have probably also heard about induced pluripotent stem cells, and this has been seen, there is epigenetic reprogramming in iPSCs, and again indicating, right. So, you need to have these epigenetic modifications that actually give this flexibility. And one question that you probably also would be wondering about, right, how do you get these different tissues, right, or different organs or different cell types within a body, right. So, if you take our examples, right, we have different organs, right, different tissues and different cell types, right. So, they actually contain the same genomic DNA, right. So, the genetic sequence is mostly identical. Okay. So, then the question is, how do they actually become so different in terms of their phenotype, their behavior, or the job they are doing right inside our body. So, this is again comes to the epigenetic modifications, because then this can actually specify, okay, which genes will be expressed in which tissues and which genes will be expressed in which cell types, or which genes will be silenced in which tissues and cell types. Okay. So, this is again determined by the epigenome or the epigenetic modifications that we talked about. So, as you probably now realize that this flexibility is also important for development of organisms, right. So, if you talk about development of multicellular organisms like us, right. So, we start from a single cell and you have this development process, right, that goes on for quite a while, right. And what researchers have seen, that you have this epigenetic reprogramming during development, right. Different marks are being added, removed, etc., and that again controls expression of different genes, right, that actually are required for at different stages of development. So, again you can do this, you can observe this very nicely if you study this epigenome along with the transcriptome. You can have something called genomic imprinting. Again, this is through epigenetic modification. So, genomic imprinting refers to parental specific gene expression. So, this is relevant for diploid organisms like us. Okay. So, where you have two copies, one copy inherited from the mother, one copy inherited from the father. Now, what happens in genomic imprinting is that only one copy probably would be expressed. Okay. So, out of these two copies, only one copy would be expressed. So, this is parental specific gene expression, and again this is specified by the epigenetic modifications. Okay. So, this is activation or silencing of genes. This is based on parental origin, but finally determined by epigenetic modifications or epigenetic marks that are present on these copies. Epigenome has a very important role in diseases. So, this has been studied now for many diseases, but a particular good example would be cancer. This has been well studied, and what we have seen is that there are substantial levels in changes in DNA methylation levels in cancer, and there are databases for that where you can actually get this data and observe these differences. And what researchers have also seen is that flexibility in expression programs actually lead to cellular transitions that can help in cancer progression or also can give rise to drug resistance. Now, as I have mentioned, right, you have the flexibility, right. So, the cancer cell can exist in two different states. One state can be expressing certain genes that actually give rise to drug resistance, but on the other hand it can have another state, right, and again this is reversible because of these epigenetic modifications, and this state can then be able to grow fast, right. So, you can have these multiple states existing within a cell population because of these different epigenetic modifications. So, because of this importance, you will find different initiatives for looking at the epigenomes and generating reference epigenomes. So, this is one such initiative, and the goal is to generate reference human epigenomes. Okay, and here are some papers and the link for the data sets that are coming out of this initiative. Okay, so, it is called NIH Roadmap Epigenomics Consortium. Please look up, and similarly you have another consortium which is looking at again human epigenome. So, International Human Epigenome Consortium, and one of the difficulties I mentioned, right. So, unlike genomes, right, these epigenomes will be very different across different cell types and different tissues, right. So, it means there would be multiple reference epigenomes, right. So, even within a single even within one species, right. Similarly, there has been lot of studies on DNA methylation in cancer, and here are again links to two databases that actually contain data on these DNA methylation studies in different cancer types, and also some normal samples, and you can clearly identify these differences across the genome. Now we come to the part for, to study the epigenomes, right. So, how do you study epigenomes? Okay. The why part is I think is clear, right, and I hope I have convinced you that this is a very important part. So, the question is now how do you study epigenomes? Okay. So, earlier methods before NGS, so they relied on mostly Sanger sequencing, right. We will discuss again these methods when we go into subsequent classes. Also PCR amplification based detection, again we will see some examples as we discuss different techniques in next class. Researchers also relied on restriction enzyme digestion. Okay. So, what you probably notice is that these are small scale methods, right. So, you cannot apply all these methods to genome wide scale, right, and most of them are localized to certain genomic regions or segments. Okay. And this all changes with the next generation sequencing methods. Now, if you wanted to do the genome wide studies, then of course, we had to rely on microarray based methods earlier, right, and this was genome wide. But then of course, we have discussed already about the limitations of microarray methods, and again the sensitivity and specificity those are much less in case of microarray based methods. And this is where the next generation sequencing methods come in, right. So, the current methods they rely on next generation sequencing based approaches. These are genome wide and they are sensitive and specific, as we will see as we go into different examples in subsequent classes. Here are the references for this class, and some of these references actually talk about this impact of epigenome, right, for disease. So, as you can see some of these references talking about cancers, right, drug resistance in cancer or in development of cancer, right. So, you can see this here in these papers. To summarize, so we have discussed about epigenome, so which is actually genome wide study of epigenetic modifications, and we have talked about different types of epigenetic modifications. So, to include right, so we talked about DNA methylations, histone modifications, chromatin remodeling and 3D genome configuration. We will talk about all these different types of modifications later on, and how we actually identify these different types of epigenetic modifications through application of next generation sequencing technologies. We have also seen that epigenome plays key roles in development, differentiation and diseases. For example, disease like cancer, this has been well studied. And earlier methods for epigenetic studies, right, so they were limited to specific segments, right. So if you talk about Sanger sequencing or amplification based methods, they are limited to only small segments of the genome and not sensitive and specific enough. If you took microarray, this is again we know the limitations of microarray methods, and this is where NGS technologies, they provide high throughput approaches, and these methods are sensitive and specific. So, in the subsequent classes we will actually see application of NGS methods for detection of these different types of epigenetic modifications at the genome wide scale. Thank you very much.